Okay, so in this second part of your lesson today, we are going to talk about the derivative of the lateral plate mesoderm, and in particular the formation of the blood vessels and the heart. So the formation of the heart and the circulatory system is actually well conserved in all vertebrates, and even before that we can find a primitive heart formation in tunic animals, which is very similar to what happened at the beginning of development of the heart in vertebrates. So we can, for example, use an example of amphibia, frogs, to study the development of the lateral plate mesoderm into the heart and circulatory system, because it is almost exactly identical to what happened in other animals, including mammals and us. The lateral plate mesoderm, here you can recognize it on both sides of the animal. Here we have the neural tube, the notochord, the somites are forming near the neural tube. Then we have intermediate mesoderm, and then we have the lateral plate mesoderm. Now, the lateral plate mesoderm is splitted dorsoventrally into regions. One region that is near the ectoderm, and this is called the somatic mesoderm. One region that is near the endoderm, which is called the splanchnic mesoderm. Now, the somatic mesoderm with the sternal ectoderm form a structure that is called the somatopleure, while the splanchnic mesoderm with the endoderm form a structure that is called the splanchnoplure. The space in the middle between the two splitted lateral plate mesoderms, so the somatic and the splanchnic mesoderm, is called the column. The column is the region that forms the body cavity. In fact, the left and right column at one point during development fuse together and they form a single cavity, not separated in left and right, and this single cavity is then separated in three by the somatic mesoderm. So the somatic mesoderm proliferate and separate the column in three regions. And these three regions, or three cavities, are called the pleural, the pericardial, and the peritoneal cavity. The pleural cavity is forming the thorax, where the lungs of the animals are situated. The pericardial cavity is forming a cavity where the heart is situated. And then we have the peritoneal cavity, which is the abdominal region, the abdominal cavity, where the different organs, including liver and pancreas, are situated. Now, the formation of the heart is very well conserved in all animals, and we can recognize the same initial development even in tunicate. In tunicate, the heart is a tubular structure and it contains only a few cells. But this initial tube that we can recognize during the development of the embryo of the tunicate is very similar to what's happened during the development of the vertebrate heart. So first of all, the vertebrate heart, it is obviously a bit more complicated than a tunicate heart because it includes many more cells and many more and different chambers. However, initially it is formed as a single tube, like it was a tunicate animal indeed. The cells that form the heart in vertebrate animals are coming from the two splanchnic mesoderms. The splanchnic mesoderm was initially separated left and right, but then they come together and they fuse together in the middle. And part of the splanchnic mesoderm is forming the heart. Not all the splanchnic mesoderm is forming the heart, but part of it. And this splanchnic mesoderm, which is in an anterior position along the anterior-posterior axis, is called the cardiogenic mesoderm. Cardiogenic mesoderm because it's the one forming the cardio. The cardio is the heart. So initially, we recognize in this cardiogenic mesoderm two regions, or if you want, two heart fields, the first and second heart field. And you can see them here in this cartoon. Now, the cell that will form the first heart field fuse together in the middle of the body, okay? And they form a tube, actually a primitive tube, which is supposedly is the primitive, if you want, remind us of the primitive heart or the heart of a tunicate. So the cells of the primitive tube, of the first heart field, are the cells that are going to form in the adult heart the left ventricle. So if here in this cartoon we can see the 4 chambers of an adult heart, of a typical arthropod, a typical vertebrate animal, typical mammals if you want. The heart is divided in 4 chambers. The 2 chambers that are posterior, if you in a posterior position, are the 2 ventricles, and the more anterior chambers are the 2 atrium. Now, the formation of the heart is peculiar because this structure with the 2 atrium on top and the 2 ventricle on the bottom is obtained after a process that is called looping. In fact, the cells that find themselves in an anterior position in the adult heart are actually developing in a posterior position in the developing heart. And only after the looping, these posterior cells that are below, they are moved on top. So the heart is formed upside down, if you want, and then it loops up. So then the ventricle cells find themselves in a posterior position, and the atrium cells find themselves in an anterior position. But initially, they are formed, if you want, upside down, with the ventricle on top and the atrium on the bottom. So here, you can follow here, I hope, from these 4 cartoons, the progression of the development of the heart. So the cells here that are going to form the left ventricles, in pink, come from the primary or the first heart field of the primary heart tube that are initially in an anterior position, but then after looping, they find themselves in a posterior position. Okay, so the cells from the secondary heart field, and the secondary heart field do not form a tube initially. They are cells that surround, if you want, the primary tube. These cells are responsible for structure of the heart that are both in anterior and a posterior end. So posteriorly, the second heart field cells are going to form the 2 atria. Now, the 2 atria, the 2 chambers of the atria, are initially formed posteriorly. You can see them here forming. And most of the cells of the atria come from the second heart field. However, some of the cells come also from the primary heart tube. But they're forming posteriorly initially, and only after looping, these posterior cells find themselves in an anterior position in the adult heart. Anteriorly, the second field cells form also most of the right ventricle. So the right ventricle is also formed mostly anteriorly during development, and only after looping, these anterior cells find themselves in a posterior position in the right ventricle here. The second heart field cells also are responsible for the formation of the outflow region of the blood from the heart. So the aorta, for example, they push the blood from the heart to the rest of the body, and the pulmonary veins, they push the blood from the heart to the lungs. So the complex structure of the 4 chambers heart and the position of the different cells change, of course, after looping. So it is only after looping that the ventricle cells, which are actually formed in an anterior position, find themselves in a posterior position, while the atria cells that are actually formed in a posterior position find themselves in an anterior position. Now, We know, or I told you, that the lateral plate mesodermal cells are responsible for the formation of both the heart, but also the blood vessel as well as the blood. But how do the blood vessels, blood, and heart are specified? The specification of which part of the lateral plate mesoderm is going to do which part of the body or the tissue, so whether it's going to become heart or whether it's going to become blood vessel or blood itself, depends on interaction between the lateral plate mesoderm and the endoderm. And in fact, one important signal from the endoderm is BMP. BMP, in particular in the anterior position, induce the formation of cardiogenic mesoderm. In the anterior position, so in the anterior lateral plate mesoderm, and here when I'm talking about the anterior, I'm talking about the anterior posterior axis. So in the most anterior position, the lateral plate mesoderm can become a cardiogenic mesoderm because of BMP signaling from the anterior endoderm that induce expression of FGF8 in a more anterior position. In the middle of the body, and here I'm talking about the middle of the body, so in an anterior-posterior way, the notochord is expressing noggin and chording, which inhibit BMP. So the notochord signal block BMP in the middle of the body. The neural tube is actually expressing wing signaling, and wing signaling are inhibitors for the formation of the heart. So because of this expression of wing signaling, the lateral plate mesoderm will become or will induce the formation of blood and blood vessel in general. And it doesn't do so only anteriorly, because in the anterior endoderm express wing signaling inhibitors like Cerberus. So Cerberus and other inhibitors expressed from the anterior endoderm block wing signaling expressed from the neural tube, so then the anterior lateral plate mesoderm can become cardiogenic mesoderm, thanks also to expression of FGF8, which is induced by BMP, also expressed by the anterior endoderm. So it is this block of wing signaling that are important, as well as the expression of BMP for the formation in the anterior position of the cardiogenic mesoderm. So the heart is probably it is the first organ that is produced during the organogenesis of the embryo or any vertebrate embryo. And it is obvious because as soon as the embryo starts to develop, you need to distribute resources around the body so it can continue and fulfill the development. Without the heart beating and therefore the blood flowing around, there cannot be this redistribution of resources, and therefore the development will block. However, the blood cannot move around without blood vessels, and the heart doesn't start beating unless the blood vessels are produced. By the way, we are going to see a beating heart in the chicken practical. We are going to see a 2 to 3 days old chicken embryo, and we will be able to see the beating heart of the embryo. The blood vessels are produced by two processes. The first process is called vasculogenesis, and during vasculogenesis, a primary network of vessels are produced directly from the lateral plate mesoderm. Then a second process called angiogenesis. During angiogenesis, the primary network is reorganized, and basically there is addition of capillaries. Capillaries are the very fine tubes, if you want, very fine tubes or networks that surround the organs and distribute resources and collect waste from the different organs. And also during angiogenesis, we have distinction between arteries and veins. A number of signals are important for the specification of cells that will produce blood and blood vessels. And these are BMP, Wnt, and Notch signaling. All of them contribute to the activation of ETV2 in the lateral plate mesodermal cells in the posterior, specifically in the posterior region. And these cells become hemangioblast. Hemangioblast cells are both precursors of blood and blood vessels. The hemangioblast can either become blood cells, and therefore they become hematopoietic cells. So hematopoietic cells are the cells that will become different blood cells. But hemangioblast can also become endothelial cells. Endothelial cells are the cells that produce the blood vessels themselves. Whether hemangioblast will become hematopoietic cells or endothelial cells depend on the level of expression of Notch. High level of Notch induce hematopoietic cells, while low level of Notch induce endothelial cells.